Good afternoon, children. Last time we had started with our new lesson, genetics, some basic fundamentals, where I had given you some definitions to write. We had written genetics, genetic engineering, then genetic counseling, certain more terms like variation, character, trait, and then a table number 1 that I had given you about inheritance in humans, where I told you character and trait. Today we'll be starting with the working of our crosses. So let us just revise from this table, children. Character is a feature of our body. For example, color of the eyes. If it is one of the expressible characters, then the trait will be the possible amount of choices available in the character. For example, brown color or blue color. Suppose eyebrows as one of the character, then whether is it bushy or thin becomes the trait. Now today, as we move with our new lesson, just to remind you, these terms you'll understand today better with the working of our crosses. For example, phenotype, genotype, the crosses, etc. Like last time we had done what is genotype, what is a phenotype by definition. Today you'll see it by working exactly, and I'm sure you will be able to understand much easily and in a better way. So what is homozygous, heterozygous, etc. So let us move to our today's learning, children. And before we introduce the terms, let us revise with a few things and certain rules that are very important in the field of genetics. For example, in drawing the genetic crosses, what we call as a monohybrid cross. Mono means one character that you're crossing between two parents, right, at a time. So that is what we'll start today with. We have sometimes to pick up a capital alphabet which represents a dominant trait, right? And sometimes we pick up a small alphabet which represents a recessive trait. Dominant means which expresses. For example, tallness. So if it is double T, it means the tallness feature in a homozygous condition. Homo means both the alleles should be the same, right? Capital and capital. Then if I write down the small alphabets, then this means a recessive trait. Recessive means it will exhibit only itself when it's in a homozygous form, like both are same. If you remember last time, children, if I write it like this, both capital, this means that the plant we are depicting would be tall. Reason? Because they are homozygous and they are dominant. If I write both different, like one capital and the other small, then it becomes a heterozygous condition, and this again will be tall, right? Because 50% of the genetic feature, it is dominant. So dominant expresses itself whether it is in double or it is in single. This is what you have to learn, children. If it is homozygous, the plant again would be tall. Similarly, if it is a heterozygous, again it will be tall. Why? Because the plant is being dominated by the dominant gene, that is T, right? But if it is something like both the alphabets that are small, then in this case, the plant would be a dwarf plant, because yes, it is homozygous, but a recessive character, the recessive allele, so it will be for the dwarfness. So one thing before we move further, a homozygous condition, you have understood, it's a double alphabet, both capital or both small, right? Heterozygous means one capital and the other small. Always remember, children, the capital is given priority and written first. You cannot write like this. See, I have already crossed here. A dominant gene is to be written first. Like this is a correct method. T followed by small t, capital followed by small. But if you write for some reason small t written by capital, children, you will be wrong. So please don't adopt this method. Initially you might pick up, sometimes by careless mistake. Don't pick up this. This is wrong. Then always remember another method, another rule. Dominant allele plus a dominant allele gives a dominant phenotype. For example, both the capital alleles T and T give you a homozygous condition like a dominant phenotype, both capitals. Similarly, a dominant, that is T capital, recessive, always remember, means a small t, and then they together give you a dominant phenotype for capital and small. But when both are recessive, that is small and small, again the phenotype would be recessive phenotype but a homozygous condition. So you saw children, homozygous followed by heterozygous and then homozygous. I think before we come to this, we should start with our working. Now these are two tables, children. You have to really learn it by heart, because without these two tables, this is for our numbering sake, table 2 and table 3. Please write it down and learn it by heart for any number of crosses that you may do. This is the dominant versus recessive trait in forms of some common hereditary traits in humans. For example, eye color. So which color is dominant? It is brown, dominant over blue. Tongue. If we can roll, we are rollers, then that's a dominant trait versus to recessive, which is non-rolling. Right-handedness dominant over left-handedness when it comes to hand use. Earlobe free is dominant over the attached lobe. Thick lips dominant over thin. Positive Rh blood group dominant over negative. Normal eye vision dominant over the color blindness. And dimpled cheek is dominant over normal. This is the category in the humans. Another table which is of counted 7 contrasting features what Mendel had picked up. 7 pair of contrasting features of garden pea, that is Pisum sativum, the scientific nomenclature. And remember, you have to underline it following the binomial nomenclature. These were studied by Mendel. So what was the character? He studied many things. He studied flower color. So the purple was dominant over white. Then seed color, yellow was dominant over green. Seed shape, round was dominant over wrinkled. Pod, that is a fruit. The green fruit was dominant over yellow. Pod shape, the inflated, that means, I'll show you here. Inflated looks like this. Whereas constricted means you can see constrictions clearly. So inflated dominant over constricted. Then we have the flower position axillary, that means between the stem and leaf, the axil of the flower, that is the angle, dominant over the terminal, which is at the terminal tip of the plant stem. And finally, the length or the height of the flower, the tallness dominant over dwarfness. So to study today with our monohybrid cross children, I'll be working stepwise and see that you follow this. This rule what you're going to follow in my working, I haven't written it already. I'll be writing with you one by one so that you stepwise understand. Please stick to this method. In your book, there's some method given like this, some horizontal laying out of flowchart, etc. But we'll adopt the method we are going to use for our overall uniformity. The example says, pure tall pea plants, that is capital allele means this is dominant. The tallness is dominant. So a cross was made, children. A cross means a fusion or fertilization or a genetic hybrid was being done with tall pea plants, capital T, right? Tall pea plants were crossed with dwarf. Now remember children, dwarf we understand, but you cannot write small d. D if you write like this, you will be wrong. Why? Because both the alphabets have to be the same. The only difference would be capital alphabet and a small alphabet. So tallness into dwarf plant again would be T and not D as I told you over here. Alright. So progenies were obtained. That means a new generation was obtained. Draw a Punnett square to show the ratios of phenotype, that means the physical observation or the character, and the genotype, which means the genetic makeup in F1 and F2 generations. Let us begin. Now, taking this here, that's already the information given in the question that pure T, capital double, was crossed with dwarf, small double T like this. Let us make them as our P1 generation. P1 means first parental generation. These are our first parents because we're going to start with the working. Now the first step over here, children, you're going to fuse the genes or the gametes that are formed. So the second step would be the gamete formation, right? On your side you can write down. And how to bring the gamete? Just bring it to their half. Half means this is double T. So what will be the half? Single T. This is double T but small alphabet. So what will be the half, children? A small t but single. Then forming the